Imagine a book that contains every instruction needed to build and run your body, your genome. It's written in a language of just 4 letters, A, T, C, and G. This book is massive, about 3 billion letters long, packed into nearly every cell. When scientists first looked at it, they had all the pages, but no order, no table of contents, just a jumble of letters. The instructions for life were hidden in there, but no one knew how to read them. It was like assembling the world's largest jigsaw puzzle without knowing the final picture. Scientists realized that if they could read this book from start to finish, they could unlock the secrets of what makes us human. The challenge was enormous, far bigger than any one person or lab could handle. It would take a global team, new technology, and years of effort. The quest to read this book became the Human Genome Project. For the first time, we had the tools and ambition to try. This wasn't just about science, it was about understanding ourselves at the deepest level. The Human Genome Project would become one of the most important scientific projects in history. The Book of Life was open, and the adventure was about to begin. Science was about to take one of its biggest leaps forward, and it all started with the dream of reading our own instruction manual. The Human Genome Project, or HGP, was a global scientific mission that began in 1990. Scientists from around the world joined forces to read the entire human genome, 3 billion letters long. Their goal, create a map of our genes and sequence every letter in order. Genes are the chapters in our Book of Life, each with specific instructions. Sequencing meant reading every letter, one by one, to create the first complete human instruction manual. The project took 13 years, finishing in 2003, and required new technology and unprecedented teamwork. Scientists worked around the clock, piecing together fragments of DNA like a giant puzzle. All the data was made freely available online, supercharging research worldwide. The HGP became a resource for all humanity, accelerating discovery and innovation. It was a gift to science and to everyone on Earth. Why go to all this trouble? Because understanding our genome is key to understanding ourselves. Many diseases are caused by tiny errors in our genes. Reading the genome helps us find and fix them. This knowledge is the foundation for personalized medicine, treatments tailored to your unique DNA. It also reveals our shared human story, tracing our ancestry and showing how closely related we all are. The Human Genome Project connects us, breaking down barriers and highlighting our common heritage. It's about building a foundation for future discoveries and better health. Reading the genome was just the first step. Understanding how it all works is the next. The project created a resource for generations of scientists to build upon. It was an investment in a healthier, more connected future. When scientists finished reading the genome, they made some astonishing discoveries. Humans have only about 20,000 to 25,000 genes, far fewer than expected, and about the same as a tiny worm. Complexity comes not from the number of genes, but from how they're used and regulated. The DNA of any two people is 99.9% identical. All our diversity comes from just 0.1% difference. This fact underscores our shared humanity. Genes make up only about 2% of our DNA. The rest, once called junk, is now known to control when and how genes are used. This regulatory system is what makes us so complex. The HGP also produced a reference sequence, a master copy for comparison. Scientists can now quickly find genetic differences linked to diseases. This has sped up the discovery of genes behind hundreds of conditions. The Human Genome Project gave us an essential tool for modern biology and medicine. It changed how we see ourselves and what's possible. The Human Genome Project's discoveries are transforming medicine right now. Doctors use genetic testing to diagnose diseases earlier and more accurately. Newborns are screened for rare disorders, allowing for life-saving treatments before symptoms appear. Personalized medicine is now a reality. Treatments can be tailored to your DNA, especially for cancer. Drug development is faster and more precise, targeting the root causes of disease. Consumer DNA tests connect people to their ancestry and provide health insights. Gene therapies are emerging, aiming to fix faulty genes and cure diseases once thought incurable. The HGP laid the groundwork for these breakthroughs. Our understanding of genetics is improving lives every day. The future of medicine is being written in our DNA. With this knowledge comes responsibility and tough ethical questions. Who should have access to your genetic information? Privacy and protection are crucial. Laws like GINA help prevent genetic discrimination. but we must stay vigilant. Gene editing technologies like CRISPR offer hope for cures, but also raise concerns about designer babies and where to draw the line. These choices require global conversations involving everyone, not just scientists. We must ensure fairness, equality, and access for all. Education is key so people can make informed decisions about their DNA. The ethical challenges are real, but not insurmountable. By thinking ahead and working together, we can use this knowledge wisely. The future of genomics depends on our choices today. The Human Genome Project was a journey of self-discovery for humanity. Our DNA reveals that we are all connected, 99.9% alike, one big human family. The project transformed medicine, biology, and our understanding of ourselves. But this is just the beginning. The next chapter is being written now. Scientists are exploring how genes interact and how our environment shapes us. Imagine a future where genetic diseases are cured before they cause harm, and medicine is perfectly tailored to you. These possibilities are within reach, thanks to the HGP. Science is a journey, and the adventure continues. The story of life is still being written, by all of us together. The best is yet to come.